Hi, class. Welcome back to the second half of today's lecture. So this part of the lecture, we're going to talk about the actual searches for life that we've done on Mars and what our plans for continuing to do those searches are in the future. So the most famous example of this is with the Viking mission. And so most of the results that we know about life on Mars come from Viking. And even though they're 30, almost 40 years old now, we are still debating what those results mean. And that's part of the story that we're going to tell today. Okay, so in the background is a artist picture of Curiosity trundling across the surface of Mars. There have been many awesome rovers on Mars, starting with Sojourner and, of course, Spirit and Opportunity. But Curiosity has always been my favorite. So Curiosity is trundling across Mars here, and this is kind of how I imagine it would be if we were hanging out on Mars watching Curiosity do its business. Okay, so let me go ahead and start some slides. Okay, did I mention Curiosity is my favorite rover? So Curiosity, you know, the Spirit and Opportunity were enormously successful. They were both there for far longer than their projected lifetimes. But they were solar powered, and so that meant they can only operate during the Martian spring and the Martian summer. But Curiosity has a nuclear power plant on it, so it roves through the winter and through the summer, and it goes all over the place and does all kinds of cool things. So Curiosity is definitely my favorite. So what we're going to do today is we'll talk a little bit about the equipment that we use to search for life. We'll talk at great length about the Viking life experiments, and then we'll mention briefly at the end what the future for searches for life will be. Okay. So the very first thing I like to point out to people, because you almost never hear it discussed, is that cameras are the absolute perfect experiment for searching for life. Okay, you almost never hear them mentioned in the context of the search for life. And that's kind of silly. So look at this picture of Mars right here. So that's a Big Joe, which I told you about. That's the rock that Viking 1 almost landed on. And when you look across the surface of Mars, your eye as a human is very good at picking out things that look unusual. You can do experiments all day long, all you want, but if you saw some evidence of life here, you know, a funny colored patch on the side of a rock, maybe it's lichen, something that looks like a pool of standing water, tracks across the surface, you know, something sticking out from behind the rock, right? You would be very good at picking that out. And that's why cameras are so invaluable on missions. You really can't send a mission to Mars without sending a camera, because even if you're letting the robot do the search for life for you, you're using the camera to pick out and use that human intuition, that human perception that you have to decide that's a better rock to look at than that one over there. Or I see a Martian hiding behind that rock, like you see in this picture. Okay, so we've certainly used the cameras on Mars to great effect. So these are all examples from the drill sites for Curiosity. So Curiosity has a very large drill on it. If we look for areas that look like they're good for sampling, we drill into it, and then we sample both below the material below the surface and above the surface. So you can see the drilling here. And so if you look at the background of all of these images, the surface soil is very different than the subsurface soil. Okay, so the subsurface soil you can see in the tailings, as we say, that are pulled up. And you can see in different places, the consistency of the soil has different behavior as well. And so we do analyses on all of these. We look at the soil behavior. If there were something in the soil that might be indicative of life, then you might be able to find it. Little structures, the blueberries that we've talked about in the past, any sort of things like that. Okay, so cameras are absolutely crucial to this experiment. So these are the kind of, these are cameras not attached to the drills. These are cameras that are either on the landers themselves or on their robotic arms. Okay, so up there in the upper left, that's the rock at the Pathfinder site called Yogi. Okay, so you can see the outline of the bear's face right there, okay? But if you look at Yogi, this is what I was telling you in the last thing, it definitely has different regions across the rock, different patches. Okay, so the top half of the rock is just a little bit different color. That's a photographic artifact. But if you look at the lower half of the rock there, you see bright patches, dark patches, gray patches, and then red patches. You see different textures. That's the sort of thing that cameras are extremely good and useful for in terms of trying to understand where to look for whatever it is you're looking for. On the upper right there is a close-up picture of a rock. Okay, and you'll notice on the top of the rock there are some interesting little structures. Okay, those are not common shapes that you're used to. They are a little bit unusual. They're each about the size of a grain of rice. Okay, so they're certainly much larger than a bacteria might have been, but they're the sort of thing that you might look for if you were looking for some kind of signature or something interesting to indicate that there was some interesting geology or some interesting influence there that was different than the surrounding environment. In the lower left there, okay, you can see one of those blueberries. So that's the kind of discovery moments where we found these things. We were looking at rocks on the ground. You see the kind of normal sand matrix, the normal kind of rocks, and someone's like, hey, what's that big round thing there that looks like a paintball, right? Okay, and it's one of these hematite blueberries. And then over here on the lower right, this is actually a micrometeoroid, okay? So this is a discovery by Curiosity. It was an odd-shaped, odd-colored rock. Curiosity also has a laser on it, and so it uses its laser to do surface chemistry on these kinds of samples, and it took a close-up picture of this, and we eventually decided it was a meteorite, okay? Okay, so cameras are extremely valuable for looking up close. We have yet to send a microscope to Mars, so we can't do the kind of up-close microscopic looking that we did like in the cross sections of the Allen Hills meteorite. But if there is any kind of macroscopic evidence of life, then our cameras may be perfectly well capable of picking it up. Okay, so let's talk about Viking. So Viking went to Mars in 1976. So both landers landed on Mars in 1976, and they operated until the early 1980s. And at the time, they were the most sophisticated scientific spacecraft ever built. They were the first landers to land on another planet. We had sent landers to the moon before. With the surveyor probes. But Viking was a lot more sophisticated. It lived a lot longer. It did a lot more. Like Curiosity, it had a nuclear power plant on it, so it could operate through the winters. But it had a variety of scientific instruments on it, ranging from the cameras, which you see right there. It had this sample arm that stretched out to the front of it. It could pivot around. It could it has a brush on the back side of it. Has a shovel on one side of it. It can dig trenches and dig up dirt. And the dirt it delivered to the soil laboratory right here. And so the soil laboratory has a variety of sifters in it. Those sifters then collect the soil and deliver it to the various Viking life experiments that was carried by the spacecraft. It had a whole bunch of other instruments as well, including weather stations and various things that it was using for other investigations on Mars. So this is a short video from Carl Sagan's Cosmos showing the operation of the Viking Arm. You can see it's on a long extendable arm there that will push down into the soil. It has a shovel on the end, which it opens up and then clamps down to keep a soil sample within it. Okay, it retracts onto the spacecraft once again. Okay, and you can see there below the shovel, there's that brush hanging down below the shovel that you can use. It turns itself over, and you see it has a sieve there to protect, keep little pebbles from falling through. But then it claps back and forth, and it drops down little bits of Martian soil onto the soil laboratory there. The soil laboratory then sifts through it again, and then delivers it to the 4 different Viking life experiments. In this picture, I'll also point out there in the background to the right and below the US flag there, you'll see a card that has a set of color bars across the top of it and a series of gray bars. And you'll see these often on NASA landers. These are basically color calibration. This is how you'll often see pictures of Mars and we'll say, this is what it would look like if you were standing on the surface of Mars looking with your own eyes. And so it's because we've calibrated it against some known color reference that we know what it looks like on Earth. We take the same picture on Mars of the same card, and then that helps us balance the color in the pictures correctly. Okay? So here's a picture of the trenches dug by Viking 1 at Chrysi Planitia. Between the two Viking sites, I think we collected 7 different soil samples, but you can see you kind of just dig these trenches into the dirt. Sometimes we dug deep, sometimes we dug shallow, sometimes they moved it and dug in a different place. But these are where the Viking soil samples came from. Give you an example of what it looks like on the surface. Okay, so what were the different experiments? Okay, so there were basically 4 experiments. Okay, so the first one was a gas chromatograph, a mass spectrometer. So basically what this experiment was doing is before we did anything to try and look for life in the soil, we wanted to know whether or not the soil itself contained organic molecules. Okay, and so what the gas chromatograph does is it just bakes the crap out of the Martian soil sample, and then we look at the chemical content and see, are there any organic molecules? Okay, and what we discovered from that experiment is that the answer is no. Okay, so we'll come back to that in a minute, because this is actually one of the points about the Viking life experiments that causes a little bit of confusion and has been much debated until the Phoenix Lander landed in just about a decade ago. Okay, so this was just to tell us something about the chemical composition of the soil, but everything after that was an actual experiment designed to look for life itself. And so basically what we were trying to do is to incite life to do something and then detect something that we could point at and say, oh, that thing that we just saw is a consequence of life doing its lifey business. Okay? Now, in the interest of not wanting to fool ourselves, we looked at what the experiments were, and we decided what the answers would be before launch. That is to say, if the experiment does this, that will be positive evidence of life. If the experiment does this, that will be negative evidence for life. We didn't want to get the experimental results back and then decide, well, does that mean that there's life or does that not mean there's life? We wanted to ahead of time say, we designed the experiment with these principles in mind, and this outcome means there is life, and this outcome means there isn't life. Okay? And we'll talk about the consequences of that decision here before the end. Okay, so the first experiment was called the gas exchange experiment. Okay, so basically what you did is you took a Martian soil sample, you removed it from the Martian atmosphere, and you replaced the atmosphere with something inert, so helium. Okay, so the noble gases, you'll remember on the right side of the periodic table, those are all inert. It's very difficult for things to incorporate them into compounds. They only appear as individual atoms by themselves. Okay, so you replace all the atmosphere there with inert helium, and then what you do is you add nutrients to the soil sample, stuff that bacteria on Earth would like to eat. Okay, and then you look for waste products that we associate with metabolic processes. You look for carbon dioxide, you look for oxygen, you look for nitrogen, methane, anything that life might give off if it consumed some of those nutrients. Okay, we did that experiment both just with adding the nutrients, but then we also added water and nutrients and looked for the waste gases again. Okay, and the outcome of that experiment was, whoa, we see waste gases that we associate with metabolic processes of life on Earth. Okay, so that's exciting because that suggests we have a sample that doesn't have any organic molecules in it. We add nutrients and stuff to the soil, and something felt good about that, consumed it, and produced some waste gases. Okay? The next experiment was called labeled release. Okay, so the basic idea here is kind of the same. But what we do is we take the Martian soil, we leave it in the normal Martian atmosphere, and what we do is in the nutrient solution that we add to the soil is tagged with carbon-14. Now, you'll remember from our earlier discussions that carbon comes in a couple of different flavors, and carbon-14 is radioactive. And so life preferentially uses carbon in different amounts. And so if the purported microorganisms in the soil were to metabolize the nutrients, that means they would metabolize carbon-14, and that carbon-14 would be expulsed in their waste gases. And so the idea here is then you look at the Martian atmosphere before the experiment. Shouldn't have any carbon-14 in it. It should all decayed away. You look at the waste gases after the experiment, and if they have carbon-14 in the gases, that means whatever was in the soil consumed the carbon-14 rich nutrients, turned it into waste gases, and released it. Okay? So in this case, the very first time we did the experiment, we saw the carbon-14. We got excited, and we did the experiment again, and then we didn't see the carbon-14. Okay, so that's an interesting, maybe it did, maybe it didn't. Okay, maybe it was the sample, maybe the first sample had bacteria in it, maybe the second sample didn't, who knows, okay? But we saw it once, and then we didn't see it the second time, okay? Now, the last experiment is usually called pyrolytic release. Now, your book calls this something different. Your book calls this carbon assimilation, but I've normally seen it called pyrolytic release. So the basic idea here is that you take your soil sample, and then you strip the Martian atmosphere away, and you replace it with exactly the same atmosphere, but one that we've constructed. And in particular, we construct it by tagging all the gases in the atmosphere that we've replaced over the soil with carbon-14 again. So the CO2 is mostly what the Martian atmosphere is. It's 96% CO2. So we replace the atmosphere over the soil with CO2 that's tagged with carbon-14. So the idea here is on Earth, plants photosynthesize, so they consume carbon dioxide from the atmosphere. And so they utilize that in their metabolic processes, okay? And so here the idea was that if there's something in the soil undergoing photosynthesis, that it would incorporate the carbon-14 in its life processes, okay? So that returned a negative result. So we didn't see anything that suggested there was photosynthetic processes incorporating the carbon-14 into the experiment, okay? So this is a very ambiguous set of results, okay? Is there life or is there not life? By our criteria set before launch, it appears there is life on the surface of Mars, okay? One of them is maybe not, but by our initial criteria, we would have said that. So it's logical to ask the question in all your reading and everything you've known about Mars, how come we don't say there's life on the surface of Mars, okay? Well, the reason is precisely because it is ambiguous. So this is one of my favorite pictures of an ambiguous picture. So you look at the right here and you can see William Shakespeare's face. But if you look more closely, it also looks like an old lady walking along the River Seine. Okay, so this is what we mean by deeply ambiguous. The Viking life experiments look like there's life, but they also look like there isn't life. Okay, so over the 30 years since Viking was landed on Mars, 40 years now, we have managed to replicate some of the results that we see on the surface of Mars with organic chemistry in the lab. Others of the results we can't. Okay. We have learned that the soil chemistry on the surface of Mars is dominated by the ultraviolet chemistry that results from the fact that it's constantly being bombarded by ultraviolet light from the sun. Mars has no ozone layer. That was something that was unanticipated, okay? The 2008 Phoenix Lander discovered there are perchlorates in the soil on Mars. That was something Viking didn't experiment for, didn't look for, and in fact didn't know. Now, perchlorates are a very strong oxidizer. They have strong, strong influences on the soil chemistry. But when we sterilized the soil before the gas chromatograph experiment, the perchlorates will destroy carbon organic molecules under those temperatures and pressures. And so quite by accident, not knowing anything about the presence of perchlorates on the surface of Mars, the gas chromatograph experiment artificially convinced us that there was nothing in terms of organic material in the surface of the soil. Okay, so all of these things have kind of been swirling around in our heads for the last few decades since the Viking life experiments. And if you ask Martian life scientists, there are some of them who very strongly believe the life experiments on Viking gave positive results for a reason, and there very likely has been or is still microbial life on Mars. Some of them take these kinds of pieces of evidence and say, nope, definitely not. And some people are still on the fence about it, okay? So this is the nature of the game in the search for life. So what should you take away from this message? So we established before launch what a yes answer would be. And that's a very important step. And I think it's one that we should always still do, precisely because we don't want to fool ourselves either way. We don't want to fool ourselves into thinking there's life when there isn't life. We also don't want to fool ourselves into thinking there's not life when there might be life, okay? The experiments came back yes, and we decided they might not mean yes. Okay, so we took our rules that we made at the beginning and we threw them out the window after we got the result back. And a lot of people find that notion very disturbing. It suggests to them that we don't know what we're doing. But in fact, what I would argue is that this means we know exactly what we're doing. This is exactly the way science is supposed to work. It's a self-correcting process. You start with the knowledge you had. We thought about everything we knew about Mars. We hadn't known about the ultraviolet chemistry. We hadn't known about the perchlorates. We did the best we can with the knowledge we had, and we set out the rules that we were going to interpret the experiments under. But then our knowledge changed. It evolved. And as a consequence of acquiring new knowledge, you update how you think the universe is working. You update your beliefs in what the laws of nature are. And that's what we mean by it's a self-correcting process. Science corrects the way we perceive the world and the way we describe the world, the way we predict the outcomes of experiments using observational evidence. And when you get new observational evidence, it has to support both your old conclusions and your new conclusions. And so you see this going on here with the Viking life experiments. The existence of perchlorates completely explains what we saw with the gas chromatograph experiments and helps explain what we saw going on with some of the other chemistry. Okay? So this is the way it's supposed to work. If you don't update your understanding of the universe, if you just rely on the rules and the knowledge that you had decades ago in science, then you're not doing science correctly. So this is the way that it's supposed to work, and you should be very happy and content that we are still trying to find answers to these questions. Okay? Okay, so for the last little bit of this lecture, let's talk about the future. So the basic problem with doing this is that while Viking and the rovers that we send to Mars are extremely sophisticated instruments, the best we can build with the best of our technology, they're still not nearly as sophisticated as a human laboratory on Earth can be. And so we've started to think about the idea of what we call a sample return mission. Can you send your robots to Mars, have them collect some soil, some rocks, interesting things that you find on Mars, and return it to the Earth? Okay? Now that is a very, very hard thing to do. Not only because you got to get the material off the surface of Mars, okay, that's hard to do. You have to get it back to Earth without contaminating it. That's hard to do. And you have to return enough of it that it will be useful to use in the laboratories on Earth, right? Whatever you bring back is going to be a limited resource. It will be the most valuable samples ever returned to Earth. And so you got to bring enough to be back to be useful, but it will eventually run out as you use it to conduct your experiments. Okay? So this is the kind of premise here of what such a mission might look like. This is a launch pad with a rocket of some sort that will carry a sample. You can see that rocket looks pretty small. It is. which means it's not going to bring a ton of samples back, but it'll bring some amount. There's a little rover there watching the launch. That rover is part of the mission. So this is a kind of notional graphic that the European Space Agency, ESA, the European counterpart to NASA, who we work with quite often, has proposed. So you can see here, you'll see ESA labels on some of the things in this graphic, and you'll see NASA labels on some of them. But basically, this is going to require several large launches. 2 to send stuff to Mars. 1 is going to send something huge and capable of collecting samples like a Curiosity rover. It will rove around the planet, collect samples, and then put them in these little canisters, these little red things that you see here. The other mission will launch to Mars, and it will leave a launch pad with the sample return rocket, and then also a little small rover. And the little small rover's job is to cruise around and pick up all the canisters that the Curiosity-sized rover collected and left behind. It will load those onto a rocket. The rocket will launch and send them back to Earth. It will be captured in Earth orbit somehow and then delivered back to Earth, where it can be analyzed in a laboratory. Okay, so this is a very notional idea for how this might go. It takes a lot of effort and a lot of engineering to do, but it is the sort of thing that we're starting to think about doing. Okay, in addition to sending humans to Mars, right? Sending humans to Mars has long been a goal ever since we sent humans to the moon. And so humans certainly can do the job that a robot could. But the question becomes, is it easier, is it cheaper, is it better to send robots, or is it easier, cheaper, better to send humans to do the job there? Okay, so that was the basic premise in The Martian, right? In The Martian, Mark Watney and all of his crewmates were on Mars doing the job that a laboratory on Earth could actually do, because they were present and the laboratory was there with them. Okay? Okay, well, that's going to conclude our discussion of Mars. As we've said in the past, there's my Martian cat wandering across the landscape there. You can see him in the background. As we've said in the past, the study of Mars could consume an entire semester if we wanted to. We've kind of hit the high points. We've talked about several things many different times. And so if you're interested in Mars, hopefully this will be a launching point for your own studies. Okay, so next week we're going to leap out past the solar system. We're going to start talking about exoplanets and finding planets around other stars, and then we'll talk about how we're going to find life or search for life around exoplanets. Okay, so I hope you all have a great weekend. Take care of yourselves, be safe, and we'll talk to you again soon.